So thank you very much, Fairly. I hope you see my screen and also you hear me.
Yes, everything is clear.
Okay, perfect. So welcome everyone. This is part 3 about dynamic imaging. So in this presentation, we will have a quick look on 4D and dynamic X-ray imaging. Let's see what it is and then have a look on a couple of examples. So if we want to go beyond the material characterization and static properties and study the time-dependent behavior of materials and dynamic processes, in addition to 3 physical dimensions, which are length, width, and depth, we also need to capture the time or the changes over time, which is considered as the 4th dimension. So this is typically referred to as 4D scanning. To investigate the dynamic processes in the pore scale or geomaterials, we need sufficient time resolution. This very much depends on the time scale of the process. In other words, how fast the process that we want to capture happens. So in geoscience, we have many different time-dependent processes that would be interesting for us to capture and study on different time scales. So for example, we have salt or mineral dissolution and precipitation, which are slow processes, while there are faster ones, such as events that occur during fluid flow, like heinous jumps, or other events like mechanical failures and fracture propagations. So we have a wide range of events in terms of timing. Here on this graph, x-axis shows the time of full scans of different scanners in seconds. And we see that in general, standard lab-based CT scans are located on the slower side of the diagram. And typically, these kind of scans, they take 20 minutes at least. It can, of course, go up to even a few hours to higher for higher qualities. So for slower processes, or when we are not very sensitive with the timing, we can use the standard lab micro CT scanning in a time-lapse fashion. This means that we do the scanning in discrete or separate time steps or lapses, and then by putting these scans next to each other, we can show the changes over time. Here in this slide, okay, yes. In this slide, we have an example of time-lapse scanning. The experiment was like this, that we had a relatively heavy crude oil sample, and we wanted to evaluate the displacement efficiency, or actually to see how the oil will be removed by injected water in a porous medium. So we prepared a cylindrical porous glass with 6 millimeter diameter and 2 centimeter height. Here you can see the horizontal reconstructed slices and the 3D rendered volume of this sample. So it was placed in a flow cell, and then we saturated it with the oil. So we pumped oil in it, and then the water was injected in the sample, and then we did these scans in different time steps after injecting a couple of pore volumes. So each scan was about 40 minutes, but since we were not very sensitive with the time, this time resolution was okay for us. Then up here on the left-hand side, I chose a subvolume of the sample. And what you see here in gray, it is the extracted pore space, which is invaded by water. And the image next to it is the same. It's just after a couple of image analysis steps, it shows that individual pores which are separated and labeled, as Ben mentioned. And here we can see them in different colors. So by doing so, we could quantify the order and the degree of invasion of each individual pore. And running it again, in each step, we can see that the degree of the more pores are being depleted as the injection proceeds. And in the very last step, then we don't see that much of displacement, and this is due to the water breakthrough and the channeling of water. So now back to this slide. We see that for capturing faster events and for higher speed scanning, lab-based micro-CT may not be suitable anymore. And typically, people they go to synchrotrons for high quality, fast scanning, or actually for capturing very fast processes. Synchrotrons, they offer very high flux and therefore very fast scanning possibilities. But the problem with them is normally the accessibility. They are not always available, and in general, they are quite expensive. So to fill the gap between the time-lapse scanning and very fast, expensive synchrotron scanning, to capture the average speed processes in the lab, a so-called dynamic scanning is developed based on continuous scanning. And as the name suggests, the scanning and acquisition is continuous and non-stop, so for a period of time. This gives us the possibility to do the reconstructions in a flexible way, and not only being limited between the 0 and 360 degrees, but every 360-degree sequence can be a full data for us to do the reconstruction and study our samples. So in this slide, I show you a comparison between the synchrotron and lab-based dynamic micro-CT imaging. Just to give you an idea that how the experiments look like while they are running, this is on the left-hand side, you can see a picture, and also I will show you a short video clip of the experiment that we recently had at the Synchrotron Center in Switzerland. So I will show you here. You can see that our core holder is located here, and then we could do the imaging with a time resolution of 1 second per scan, 2.5 micrometer voxel size, and the acquisition is 180 degrees back and forth. And we can see, so normally flow experiments, they are quite complex, and there are a lot of add-ons and sensor attached to it. And it is always a challenge to see, to set the tubings and flow lines in a way not to get bent and twisted. And you see that here during the fast scan, the tubing or the flow line is moving quite a lot. So on the right-hand side, this is a lab-based setup, which is actually available in Excite network as well. This is for dynamic imaging. With this setup, we can do the scans down to 12 seconds per scan, and then the acquisition is 360 degrees rotation. And the very, very big advantage that we have here is that instead of turning the sample, and as I mentioned, that flow experiments are quite complex, and it is very difficult to normally rotate them. But instead here, the rotation, the source and detector, they rotate. It is just like medical CT. And this gives us a very high degree of stability during the experiments for us. So in this slide, just to give you an idea about the quality differences, I show you two reconstructed images from our porous medium in the lab and one from synchrotron. So on the left-hand side, you see an image from a 12-second scan in the lab, in which we have quite a bit of noise, but still in some circumstances, it is enough for us to study the dynamic processes. And then if we increase the time, scan time up to 40 seconds, then the quality becomes much, much higher. And on the right-hand side, when you see that the quality that we can get from synchrotron in a 1 second scan. So now some examples of dynamic lab-based scanning. The first one is a dynamic visualization of pore scale drainage behavior. So here we capture the fluid distribution over time in Venttimer sandstone, and the sample size is more or less the same as the one that I showed you before. It's a 5 millimeter diameter with a height of 17 millimeter. And up here we have a reconstructed horizontal slice of the sample. And this 3D visualization on the left-hand side is a subvolume of the marked area over here that we can see on the slice. So In this 3D volume, we see the grains in gray and the invading oil phase, which is artificially colored in red for a better visualizations. The sample was saturated with water at the beginning, and then the oil was pumped into the system. The scans, they were done in 12-second measurement time for 5 minutes with a voxel size of about 15 micrometers. And then here it is visualized in every 4-second step. So scanning was done without interrupting the fluid injection and without stopping the experiment. So what we can see from the data is that the pore scale displacement processes are happening in rapid interfacial jumps, which is called Haynes jump. Other than the visualization of the dynamic process, we can also implement algorithms to define and locate individual pores. And so, for example, here the pores, they were identified and labeled, and then based on this labeling, we could analyze the invaded pores, right? And if we have a closer look over here, we can see that at the time of 136 seconds, this pore, which is located in the middle, is still empty, and then in a few seconds, the Haynes jump happens, and then it becomes filled with oil. And then in another 4-second step, then it becomes connected to the oil, which is located in the neighboring pore. So another example, we have, again, a sandstone rock sample. So this is a vertical slice view of the sample. It was first saturated with water, and then the oil was injected in the system to see how the drainage process occurs. And here in this visualization, again, water was injected to see now the pore filling dynamics in a mixed wet system. So oil was decaying and water was containing potassium iodide as the doping agent. This is why that here we see the water as light gray, and the darker gray is grain, and the dark phase, or like the black phase, is oil. So if I run this one, then we see the pore filling dynamics in different pores. So for example, in pore number 1, it happens quite fast, and then in pore number 2 and pore number 3, it is much slower. So this can help us to, for example, find out that these pores are mostly isolated by micropores, and these micropores, they limit the fluid supply, and therefore it slows down the pore invasion dynamics. Another example is about solute transport in carbonates. So in this example, we have a limestone sample, which is already saturated with water, and then injecting water, which contains 10% of cesium chloride salt in it. We want to see how this salt spreads in the porous medium and even in individual pores. So this salt, cesium chloride, it has a high attenuation coefficient, and its concentration is reflected on the recorded gray values. It is injected from, well, actually this cesium chloride solution is injected from below, and fast scanning is done like every 12 seconds. And then by analyzing the recorded data and gray values, we can quantify the concentration of cesium chloride and its changes as a function of time in each individual pore. Here, the change in concentration is color-coded, and you can see, for example, in these pores, which are located on the left-hand side of the sample, the concentration of cesium chloride increases faster than the other pores. Another example, we have fracturing of carbonate rock under compression. So in here, we have a limestone sample, which is under compression, and we want to see that how fracture forms and propagates in the sample. So dynamic imaging and continuous acquisition not only help us to capture faster processes, but it is also very, very useful to capture the moment of interest. So actually to capture the events that we cannot exactly predict when they will happen. So for example, here it would be very, very difficult to capture the moment at which the fracturing happens with the time-lapse scanning. So we could easily lose it. But with continuous scanning, we can just easily monitor and capture those moments that we are interested in. We can also like reconstruct exactly the moment before the fracture, the moment after it, and to see exactly what is happening in between. So even if this time resolution in the laboratory is not enough for us, then we can go to synchrotron, of course. But still, these lab experiments are very, very valuable for preparation for synchrotron experiment. And they help a lot to know what we are expecting. And then therefore, we can design our experiments in the best way possible. So the very last example that I show you here in this presentation is a very, very new data. It is still unpublished and it is under study. So by using FastLab-based imaging, we can perform particle velocimetry in microscopic pores of porous material. Here, the experiment is like injecting these tiny particles, and then by fast and dynamic imaging, we could trace them down and see how they work. And this would be a very, very valuable data for further simulations. So far, we talked about the whole CT scanning and the dynamic scanning. So it not only help us to directly see the processes in very small scales, but it can also be used to extract and model pores and throats and set up pore network models. So for instance, here you can see on the right-hand side such a pore network model in which the pores are represented by red spheres and are connected through channels, which is extracted from the CT data on the left-hand side. These kind of models, they can be used to simulate fluid flow in porous media, and dynamic imaging, which we obtain from the experiments, are very valuable to validate the models here. So for example, here on the right-hand side, it is the drainage example that I showed you beforehand. And then on the left-hand side, it is simulated via these pore network models. And by comparing these two and then adjusting the parameters, we can validate our model by the data that we get from the experiments. So this brings me to the end of the presentation. I would like to thank you all for your attention, and if there are any questions, please feel free to ask.